Thank you very much. So we'll get straight on to the two longer talks, and we can have questions after each of the talks. So first up is going to be Agata Dziegiel from Quadram Institute Bioscience. So Agata, whenever you're ready, share screen and...
Thank you, Nicole. Great, thank you. So my name is Agata, and I'm going to be talking about our investigations of Campylobacter diversity in retail chicken. So as we know, poultry is the main reservoir of infection for humans, and this largely includes chicken meat. And mostly Campylobacter jejuni and coli are implicated. And what we know about Campylobacter diversity on the farm is that there is evidence of host generalist and host specialist lineages, and there are a wide range of these. Although in individual flocks, a small number of these or even individual lineages tend to persist. So this is a minimum spanning tree of all the chicken meat and offal isolates in the UK available from PubMLST. And if we look closer, we can see that these host generalist and host specialist lineages that we see on the farm are also represented in chicken meat. So what is happening with chicken meat? There is evidence of cross-contamination of meat during processing, and this may lead to an increase in diversity of Campylobacter. So this is a figure that I pulled from a paper looking at Campylobacter jejuni subtype richness between different stages of the poultry production chain. So we can see that there is an increase in Campylobacter jejuni diversity between the barn and the abattoir levels, and again between the barn and retail, but there is no significant difference between the abattoir and retail. So we should be able to investigate this Campylobacter diversity at the end stage of the poultry production chain. However, some of the typical laboratory conditions that are used can prevent us from being able to isolate some of the Campylobacter species or sequence types. So the purpose of this set of experiments was to manipulate a number of culture conditions and perform whole genome sequencing to investigate the species, the sequence types, and within lineage, pairwise single nucleotide polymorphism differences to begin to understand the true diversity of Campylobacter in chicken meat. So this is the workflow that was followed. So this outlines the typical method and then the methods that were manipulated. So we did direct plating versus enrichment. We used two enrichment temperatures, 37 degrees and 42 degrees. We used two different enrichment broths, Bolton broth and cat broth, and two plating media, MCCDA and unsupplemented MCCDA. So the purpose of this was to maximize the growth of some of the potentially more antibiotic sensitive isolates. And following isolation, we performed DNA extractions and whole genome sequencing. And the genomes were subjected to bioinformatic analysis. So for the purpose of this, we were interested in classifying the species and the sequence types of the isolates and then performing recombination analysis and pairwise SNP analysis of individual lineages in the samples. So from the initial 15 samples studied, 12 of these were culture positive for Campylobacter, and we obtained 167 isolates in total with a median of 8 per chicken sample. In some cases, we saw evidence of multiple species and even sequence types being present in a single sample. So in 2 cases, we had 2 species, and these were either Campylobacter jejuni and coli or jejuni and larvae combinations. And in 5 samples, we also had 2 sequence types of a single species. We also saw that conditions were important as well. So for example, Campylobacter jejuni SD464 was only isolated at 42 degrees and Campylobacter larvae only at 37 degrees. We also saw that direct plating was important for the recovery of an unknown Campylobacter jejuni sequence type, and I'll come to this in a second. And interestingly, we saw that Bolton broth enrichment yielded a smaller number of sequence types compared to the alternative broth condition. And generally, we saw that low within sample pairwise SNP differences were evident between isolates of the same sequence type. So in terms of the species split, we obtained 167 isolates, from which we obtained 164 high-quality genomes. Almost 80% of these were Campylobacter jejuni, 11.6% were Campylobacter coli, and the rest were Campylobacter larvae. So if we look at the Campylobacter jejuni genomes first, we identified 10 sequence types, 2 of which were unknown or novel sequence types. So this unknown sequence type down here was identified through direct plating only. And I also mentioned that some of the samples contained multiple sequence types. So for example, in one sample, this was SD5136 and SD257, and another ST6175 and ST464. So we can see that there is within sample diversity, but within the individual lineages, there was a small number of differences in terms of pairwise SNP differences as indicated by these ranges. In terms of Campylobacter coli, we identified 3 sequence types, one of which was an unknown sequence type, and this sample also contained ST1096, and another Campylobacter coli positive sample contained ST1595. And for Campylobacter lari, we only identified 1 sequence type in a single sample, and again, there wasn't much difference between the isolates here. So in terms of the significance of these findings, we saw that in some cases, multiple species and even sequence types were present in a single sample. And this further evidences cross-contamination with different sequence types and species. We cannot determine at which stage this has occurred. It could have occurred at any point of the meat processing stage, or even before, or even at retail level. And we also saw that some of the traditional isolation methods may not always yield a representative Campylobacter population. In particular, we saw that Bolton broth enrichment had a much lower sequence type yield compared to the alternative broth type. And we saw that direct plating may be important for the isolation of some sequence types, although this is just initial data, and we are looking further into whether these isolates can be isolated in the broths as well. We also displayed that multiple isolates should be taken to assess diversity, because if fewer isolates had been taken, we may not have been able to identify some of the sequence types or species. And this is important for source attribution and outbreak models to determine where infections are coming from. And in terms of the next steps, we are looking at incorporating alternative enrichment broths into the workflow to see whether there are any additional sequence types or species that can be identified in comparison to the traditional method. We're also expanding the sample set, and we'll be performing further diversity analyses looking at hypervariable regions and plasmids. So with that, I would just like to thank my supervisory team and the Mather group at the Quadram and Midwinter at Massey University for their expertise and helping out with these experiments, and also the funding bodies of my project. Thank you very much.
Thank you very much, Agata. So there's some conversation that you might want to follow up on. Others can pick up on Discord with regard to where some of these other strains might be coming from, whether they're from the fecal content, whether they're getting onto the surface of the bird, or whether they're coming from cross-contamination. But there's been a couple of direct questions. One relates to the use of CCDA. So Alexandra says that they have lots of problems in their lab using CCDA with other bacteria coming through as well. They've switched to now using Butzler. Do you get any culture problems with MCCDA?
So there's not been a huge amount of problems. There is some contamination that we've seen, but this is why we're also exploring alternatives of broth and plate combinations. Because I know that particularly in Bolton broth, cefoperazone is also incorporated into that, as well as into MCCDA. So we are going to be looking at alternative broths to see whether we can knock out some of this contamination earlier on. Yes, we are also going to probably be considering alternative plating types as well.
Okay, thinking about the number of samples, Albert's asked, based on the pilot study of 15 samples, how many colonies do you have to pick per sample per method to get all clones? 48 is a lot.
Yes, so at the moment we have been only doing what has been practically feasible, because there is a lot of sort of combinations of the methods. So we took 4 from each condition, but in total up to 48 per sample. So the hope is that I can analyze how many isolates I need to be able to determine whether I'm losing anything or not picking something up that's important, to see if I'm still sampling the diversity of the sample.
Okay, thank you. And finally, we have Tag says, fantastic talk, really interesting to see the level of diversity in individual samples. Any idea whether these included strains that were resistant or sensitive to different antibiotics?
Yes, so I've done some preliminary AMR analysis on these. There hasn't been any resistance to macrolides at this stage. I've mainly just seen sort of oxygenes and some aminoglycoside resistance in a small number of samples, but yeah, I'm still in the process of analysing those results.
Okay, thank you very much.
Thank you.